Hello. Today I'm going to share with you some of the progress that my lab has made towards quantifying proteins in single cells and some of our ideas of how these data might be used for spatial proteomics. In particular, we are interested in combining the high spatial resolution of imaging methods, whether that's imaging messenger RNAs or proteins in tissues and with subcellular resolution, and the depth of profiling power of omics approaches. In particular, we would like to use imaging approaches to establish the profiles of spatial marker genes that then can be used for either sorting single cells or computationally aligning them so that we can computationally reconstruct 3D single cell resolution atlases. And in particular, one project where we pursue this approach is funded by the Zuckerberg Initiative in collaboration with a number of colleagues listed below. Now, when colleagues have pursued a similar approach to measure both RNAs and proteins, they have found that there are notable differences in terms of the most abundant proteins and messenger RNAs that are found in human tissues. And of course, that comes as no surprise to many of us who have long noted that there are substantial differences between protein and RNA abundances measured in the same tissues. In particular, here on the left, if you look at the abundance of p53 transcript and protein, you see that across human patient samples, the correlation is relatively low, which in this case, of course, is related to p53 being regulated primarily via degradation. The RNA is constitutively expressed, the protein is more or less constitutively synthesized, and then the abundance is determined by the degree to which the protein is regulated, which of course explains why the correlation between the protein and RNA is only weak. And we found a relatively similar type of dependence to apply to many other human proteins, here shown with the cartoon to the right, when we analyze the draft maps of the human proteomes. In particular, we find that messenger RNAs are better at predicting overall abundance of a protein, whether a protein such as gene 5 is going to be highly abundant at both protein and RNA level, or lowly abundant, such as gene 1. But the ability to predict changes across tissues is much more limited. And then furthermore, as we are interested more towards function, protein interactions and modifications, post-translational dynamics, and so on, RNA is increasingly less and less informative. So these examples motivate the need to measure proteins in single cells, which of course has been long appreciated, and there are many approaches to do that, traditionally mostly based on affinity reagents, and more recently, there have been a number of groups, including my lab, that have aimed to use mass spectrometry to increase both the specificity and the depth of single cell protein analysis. We were concerned about pushing the frontier of affinity reagents such as antibodies because of these estimates published a few years ago in Nature that annually we waste about $1 billion on affinity reagents and antibodies that do not have the desired specificity. And therefore, we wanted to pursue mass spectrometry approaches, being particularly encouraged by this observation that on average, proteins are much more abundant than messenger RNAs. That is, for each messenger RNA in a cell, there are about 1,000 protein molecules. And this observation is very important because it raises the possibility not only to detect proteins, but also to quantify them in single cells. So it is, we might be able to sample enough copies of proteins so that we have relatively low counting noise, as shown here with this estimate based on the Poisson distribution modeling our sampling. Now, this slide is half a minute introduction to liquid chromatography, tandem mass spectrometry approaches to identify and quantify proteins. Proteins are separated either using electrophoresis or chromatography, in this case chromatography, and then in ionized, injected into the instrument as ions, and individual ions as individual clusters of ions corresponding to a particular peptide, as shown in panel B, are being isolated and broken down into fragments, and the fragments used to determine the peptide sequence. And the key part of the simple description here is that while we can determine the mass overcharge ratios of tens and hundreds of thousands of ions in a single run corresponding to peptides, this information alone is not sufficient to determine peptide sequences. We have to detect enough peptide fragments from each peptide to determine its sequence, which is this information here at the bottom panel. And some of these peptide fragments are produced very inefficiently, which becomes a substantial problem for single-cell protein analysis or any other kind of analyzing minute amounts of proteins. So to mitigate this problem, my laboratory developed and introduced the concept of isobaric carrier, where we label the peptides of small samples, such as single cells, with isobaric mass tags. And we label also a small carrier sample, which can be the peptides from a small bulk sample of single cells, again with a different isobaric tag. All of these labeled samples are mixed because the tags have the same weight. A particular peptide with a particular sequence derived from all of the different samples is going to appear as a single cluster of ions in the instrument. Then that cluster is isolated, fragmented, and upon fragmentation, the mass tags break in a way that produces distinct reporter ions or barcodes of a kind, if you will. And based on these reporter ions or barcodes, we can quantify the relative abundance of the peptide across all samples. And based on the peptide fragments, we can determine the sequence of the peptide. And specifically, what's very helpful here is that these peptide fragments in this experimental design using the isobaric carrier concept, the fragments are generated from the pooled amount of the peptide derived from all of the small samples, e.g. single cells, and from the carrier material, which provides enough fragments to determine the sequence of the peptide and increases, in many cases, enough fragments, not in all, and increases our ability to assign confident sequences. Now, does it work? The first definitive evidence that we had came 4 years ago when we compared the abundance of fluorescent proteins measured on a FACS sorter, as shown on the x-axis here, and measured by mass spectrometry on the y-axis. And the correlation at the time encouraged us that this approach can be further refined and improved. And indeed, over the last 4 years, we have substantially advanced the accuracy and the throughput of these type of measurements. Currently, one of the most widely used pipelines in my lab to do this kind of analysis that has been increasingly adopted by others as well is illustrated here with this diagram. We start by isolating single cells, typically by FACS sorting them into a 384 well plate. Then we lyse them by a freeze heat cycle, which MPOP, lysis method that we specifically optimized for this type of analysis. We digest them to peptides. We label the peptides with isobaric mass tags. You can think of this as barcoding. We mix the samples. We analyze them by liquid chromatography tandem mass spectrometry. And then we use a number of software tools to extract the data, many of those developed in my lab, so that we can optimize the parameters of the analysis and we can enhance data interpretation. Recently, we have also developed new methods for sample preparation, such as a droplet-based method that allows to do the sample prep in just a few nanoliters, here shown on the left. And this improved sample preparation also results in higher quality protein quantification, as shown here with this example of separating a few hundred HeLa cells in Q937 monocytic cells analyzed with this approach, where we cannot only see the differences between cell types, capturing 81% of the variance in the data, but now we can begin to see a lot of differences in heterogeneity within a cell type due to protein-protein interactions or the cell division cycle, and you can read about it in the preprint here reference at the bottom. Now, to illustrate the capabilities of the single-cell proteomic technologies that we have been using over the last couple of years, I'll introduce this example of analyzing the emergence of heterogeneity in macrophage populations, which is basically motivated by this question: are macrophages differentiated from clonal cells, a cell line, going to be diverse or similar? Macrophages are interesting cells because they have very diverse phenotypes in our tissues and organs. They can be either pro-inflammatory or anti-inflammatory, and we wanted to see if some of that polarization may emerge even in the absence of external cytokines. The experimental system was very simple. We in vitro differentiated monocytic cell line to macrophage-like cells and and, uh, and monocytes. And then we sampled cells to analyze either by mass spectrometry and quantify their proteins, or to analyze by 10x genomics. We applied this pipeline that I introduced a couple of slides ago. And then we just projected the data into the axis of their principal components. And the first thing that we can see is the definition of two clusters that correspond to the two cell types present in our mixtures: a more compact cluster of monocytic cells, and another more diffuse cluster in blue of the macrophage-like cells. In this case, the color coding here is based on the known cell type of the origin of the cell. So we kept track of which cell is which during the sorting procedure, but we need not have done that, because if we were to color code the cells based on monocyte markers or macrophage markers, we again can recapitulate the identity of the clusters. You can also see potentially some heterogeneity emerging here in the case of the macrophages, which we're going to examine in more details in the subsequent slides. So if we look at the genes, at the proteins that distinguish the monocytes from the macrophages, we see many of the expected functions being represented, such as protein synthesis and proliferation for the monocytes, and cell adhesion and immunology-related functions for the macrophages, which is just a validation of the approach, nothing unexpected here. The more interesting observation is that if we look just within the macrophages, we still see a gradient of variation of heterogeneity, and this gradient is something that we did not know to begin with the experiment. We had a more or less clonal population of monocytic cell line, differentiated in environment that was as homogeneous as we could make it, without polarizing cytokine, and nonetheless we see that heterogeneity emerging, and that heterogeneity we wanted to explore what might underlie it. So we looked at the functions of the proteins that are more abundant on the left or on the right of this cluster. And we found, as indicated below this panel here on the bottom, that there is almost perfect correlation to the abundance of genes that were previously identified as being M1 or inflammation polarization specific markers, or M2 and for alternatively activated or anti-inflammatory macrophages, suggesting that this propensity of macrophages to differentiate into pro and anti-inflammatory cells is inherent to them. Now, I want to come back to this point of our aspiration not only to detect, but to quantify proteins. Are we succeeding in this? To answer this question, we quantified the copy numbers of proteins and messenger RNAs detected for genes that were quantified both at the transcriptome and both at the RNA and at the protein level. And we found that for messenger RNAs, there are some genes from which we get hundreds of copies, but for the majority of genes, we have only a few copies of messenger RNAs, which corresponds to relatively high counting error, as shown here on the top. While for proteins, we sample about 20-fold more copies, and then for messenger RNAs, which corresponds to the smaller counting errors. Accordingly, with the better sampling, we see that the relative quantification of proteins is quite reliable and very similar between the single cells averaged in silico and bulk relative protein levels measured by conventional well-validated methods. We took advantage of having paired RNA and protein data from the same system, and we wanted to analyze which genes have similar behavior at the protein and at the RNA level, and which don't. So we performed correlation vector analysis based on which we identified two clusters. Cluster 1, composed of over 1,000 genes that have at least qualitatively similar profiles at the RNA and the protein level, and cluster 2, composed of genes that show the opposite behavior at the RNA and the protein level. And then we looked at the functions enriched in cluster 2 and found that many of those functions are related to cell signaling, such as RAB GDPase activator activity. We also performed joint dimensionality reduction of our dataset, which resulted in two clusters corresponding to the monocytes and the macrophages that co-clustered between the RNA and the protein cells, but the protein cluster was tighter, as shown here with blue and red, as opposed to the more diffused cluster based on the RNA data, suggesting that there is potentially less technical and biological variability in the protein data as opposed to the RNA data. We also wanted to evaluate the degree to which biases and artifacts contribute to this clustering. So we color-coded each cell based on the biological replicate and the technical replicate of the particular experiment, and we saw that there wasn't strong correlation between the coordinates of the cell and the replicates, suggesting that these biases are small and have been further reduced by the data analysis pipeline. And then we are interested in using this type of data, including spatial data with the spatial coordinates, to infer gene regulatory interactions, such as using the joint distribution between protein abundances to infer how which proteins interact together. And this is an old aspiration, an old idea that has been articulated, but there hasn't been appropriate data for the most part to apply widely. So we wanted to try to perform this kind of analysis with our joint RNA and protein profiles of the same system. So we used common principle component analysis to order cells analyzed by RNA sequencing and mass spectrometry. And here is an example of a gene, NCL, which is relatively highly abundant and changes in the same way at the RNA and at the protein level. Then another gene, RHOC, again, in group 1, shows similar behavior at the protein and the RNA level, but it's less abundant, and you can begin to see the discreteness of detecting 1, 2, 3 RNAs per cell, which is not reflected in the more continuous protein measurement, reflecting the ability to capture more copies. Now, more interestingly, if we look at p53, the tumor suppressor protein, we find that the RNA and the protein levels are not strongly correlated, which perhaps reflects the regulation of p53 by protein degradation. And then we wanted to go one step further. We wanted to ask whether we can use the abundance of p53 at the RNA or at the protein level to predict the transcription of its target messenger RNAs. And we found that the messenger RNA for p53 contains no such information, while the protein abundance of p53 was inversely correlated to the repressed targets, as one would expect, suggesting that there is some information for the regulatory interaction being captured in the protein abundance of the transcription factor. And of course, this is incomplete because our data at this point do not capture post-translational modifications and localization of P53, which may in part account for the for not seeing the expected regulation for the activated targets. So with that, I would like to thank the highly collaborative team that has made this work possible, and in particular to Harrison Specht, who was the lead student in the projects that I shared with you today. I also would like to thank uh, our funders, especially the NIH Directors Award, for giving me the freedom to pursue this research, and the Paul Allen Distinguished Investigator Award that is supporting our efforts to further advance these technologies.